Biochemistry 401 Lecture 34. Today we're going to talk about nucleotide catabolism. We're also going to discuss DNA structure, including A, B, and Z DNA, and we'll talk about histones and higher order structures. So let's get started. The breakdown of nucleotides relies on three major classes of enzymes. These are nucleotidases, nucleoside phosphorylases, and phosphoribomutases. Nucleotidases break down nucleotides to nucleosides by removing the phosphate groups. Nucleoside phosphorylase removes the base from the nucleoside to yield a free base plus ribose 1-phosphate. Phosphoribomutase isomerizes ribose 1-phosphate to ribose 5-phosphate, which is an intermediate in the production of PRPP. And so ribose 5-phosphate can be recycled to make more nucleotides. Once the base is released, it can be broken down. We're going to look at the catabolism of purines. The strategy is this. First, convert both AMP and GMP into a common intermediate, xanthine, and then convert that into urate for excretion in the urine. We're going to start with a nucleotidase that's going to cleave the phosphate group from the nucleotide AMP to form adenosine, the nucleoside. We're then going to release the amino group. We're going to use adenosine deaminase to do this, and we end up with free ammonium and inosine. And the next thing that we're going to do is we're going to use a nucleoside phosphorylase to remove the base hypoxanthine from ribose 1-phosphate. We're then going to use a very important enzyme called xanthine oxidase. This is going to introduce another ketone group in hypoxanthine to make xanthine. There's one less oxygen in hypoxanthine than there is in xanthine, and that's a good way to remember the difference between these two intermediates. And so what we're going to do is we're going to add oxygen and water, we're going to make peroxide, and we're going to make xanthine. This is a common intermediate in the breakdown of both GMP and AMP. We're going to use xanthine oxidase again, and this time we're going to make another ketone group in another position. We're going to end up with peroxide again and uric acid. This is going to become deprotonated to form urate, which is the conjugate base. And so for humans, this is where it stops, with urate. Most mammals turn uric acid into allantoin, and then this is released in the urine. Fish break down allantoin even further and release ammonia into the water. But for humans and great apes, the catabolism of purines stops at urate. This is because we've lost the uricase gene, and so we cannot catabolize urate to form allantoin. And for this reason, we excrete urate in the urine. Now the thing is, urate actually circulates in the bloodstream at fairly high concentrations. Humans have an even higher concentration of urate than other primates, and this is thought to have given us an evolutionary advantage. This is because urate is a very potent antioxidant. In fact, it's just about as powerful as ascorbate vitamin C. And so it's thought that perhaps the loss of uricase counters our loss of the ability to make vitamin C. Urate is present in high concentration in the blood, and it's really close to saturating concentrations. It can actually precipitate out as sodium urate crystals. These are shown in polarized light here, and they look like little needles. You can imagine if these precipitated out of the blood into joints or into the kidneys, that it would be very painful, and this is true. This causes a painful condition called gout, and persons who have overly high concentrations of urate can also have kidney stones. Treatments for gout are to change the diet to decrease the amount of foods taken in that have high purine concentrations. Believe it or not, one of the first things to go is beer, and shellfish, and spinach. Gout can also be treated with medicine. Now, allopurinol is used as a competitive inhibitor of xanthine oxidase. It inhibits in two places, both in the production of xanthine and in the production of uric acid. And so in this way, it blocks both the production of uric acid from AMP and also from GMP. Another problem that's associated with the catabolism of purines is something that's called severe combined immunodeficiency syndrome, and this is caused by a defect in adenosine deaminase. And so the deamination of adenosine to make inosine is deficient, and for this reason there's a roadblock. Adenosine will build up and AMP will build up. Because of the ADA deficiency, the road from AMP and adenosine to inosine is blocked. Therefore, these intermediates, AMP and adenosine, will feed into the production of ATP. Now we know that ATP is a positive regulator of ribonucleotide reductase. And because we have an overabundance of AMP and adenosine above normal levels, these will be used in order to make DATP, and because of the rise in levels of DATP, this will inhibit ribonucleotide reductase and will inhibit the production of deoxyribonucleotides as a whole. And this has a profound effect on one of the organs in our body especially. This is the thymus. The thymus is an organ that sits in the middle of the chest, just beneath the breastbone. It encircles part of the trachea. This is an organ that is responsible for the maturation of thymic cells, T cells, which are a primary cell involved in the immune response. As you can see from the slide, at birth, the thymus is about 15 grams, and it increases in size from birth to puberty, more than doubling in fact. Then there is a general decrease in size, until by 70 years of age, it's only about 5 grams. It just about disappears. The mass, in fact, of the thymus at this point is mostly adipose and connective tissue. Now, thymic tissue is especially sensitive to decreases in adenosine deaminase activity, and this is because the thymus generally has a great deal of adenosine deaminase activity. In fact, if you look at the activity of adenosine deaminase per milligram of protein, the levels of adenosine deaminase activity in human tissues taken from surgeries and postmortems on children that were up to and including 1 year old, as you can see, the greatest nanomole of adenosine activity per milligram of protein is seen in the thymus. By far and away, this is true. As we saw, the thymus should increase in size in children. This is why a loss of ADA hits the thymus hard. It causes severe combined immunodeficiency syndrome. Nearly half of all cases of SCID, in fact, are caused by ADA deficiency. This is because the maturing thymic cells, T cells, in children are especially sensitive to ADA deficit. This is because cell division is decreased because of a lack of deoxyribonucleotides for replication and also for DNA repair. Incidence of apoptosis, programmed cell death, is increased. For this reason, these children have a compromised immune system and are highly susceptible to infection. In the tissue preparation shown in blue, we see mouse thymus. On the left, we see normal thymic tissue. And on the right, we see thymic tissue in a mouse homozygous negative for ADA. Because adenosine deaminase is deficient, there is a reduction in the number of cells in this tissue and an increase in apoptosis. Those cells that are apoptotic are shown in red. And you can see that there's an increase in apoptosis in the ADA deficient thymus. The treatment for SCID these days is first chemotherapy to destroy the bone marrow, followed by bone marrow transplant from individuals that have functional adenosine deaminase. So now we're going to shift gears and we're going to talk about DNA structure and the central dogma briefly. The central dogma states that heritable genetic information is stored as DNA, and this genetic information is passed on to a daughter cell through the replication of DNA. The DNA is transcribed to make RNA, and this includes messenger RNA, transfer RNA, and ribosomal RNA. The messenger RNA is translated to form proteins. The structure of DNA is shown here. 2 strands of deoxyribonucleic acid wind around a central axis. The bases lie approximately perpendicular to the central axis. The bases are found on the inside, and the deoxyribose phosphate backbone is located on the outside. These two strands of DNA bind to one another primarily through hydrogen bonding, also somewhat due to hydrophobic packing between the base pairs. Now there are three basic configurations of DNA: aDNA, bDNA, and zDNA. aDNA and bDNA are both right-handed helices. However, zDNA is a left-handed helix. aDNA is short and broad, with about 11 base pairs per turn. It's usually seen in DNA-RNA hybrid molecules, and it's also seen in RNA-RNA duplexes. bDNA is also a right-handed helix, but it's longer and thinner than aDNA, with approximately 10 base pairs per turn. This is the type of DNA that is found in most DNA-DNA duplexes. The predominant structure of our DNA is bDNA. zDNA is the only left-handed helix. It's long and thin, and it has 12 base pairs per turn. It's found in DNA-DNA duplexes, but in special duplexes, usually in DNA regions that consist of CG repeats. And it's thought that this type of DNA serves a purpose in gene regulation. The path of the backbone traveling leftward is shown here. Now, most DNA under physiologic conditions is in bDNA conformation. The differences in A and B DNA arise from differences in ribosyl sugar pucker. This is whether carbon 2 or carbon 3 is out of the plane of the other ring carbons. The differences in B and Z DNA arise, as I said, from differences in local base sequences, where runs of D, CG can favor Z DNA conformation. Here we see the differences in sugar pucker. In the C3' endo, seen in the A form, the C3 carbon is above the ring, whereas in the C2' endo, the 2' carbon is above the ring. And this is usually seen in the B form. And so, depending on whether the C3' or the C2' carbon is above or below the ring, helps to cause a conformational change in the DNA. Do you need to remember which form is seen in the A form or the B form? No, you sure don't, but you should know the general concept. And so there are 34 angstroms for every 360-degree turn in bDNA, and there are approximately 10 base pairs in each turn. Therefore, there are about 3.4 angstroms between adjacent base pairs. bDNA is a hydrated molecule. This is generally the conformation of our DNA in our cells. Each base is tilted slightly from perpendicular. Now, as you know, hydrogen bonding occurs between guanine and cytosine and adenine and thymine, with 2 hydrogen bonds between A and T and 3 hydrogen bonds between C and G. The bases are in the ketone configuration and not the enol configuration. Now, because pyrimidines always bind to purines through base pairing, this allows the DNA to have a consistent diameter, which is important. DNA has a major groove and a minor groove, as one strand winds around the other. The minor groove side is shown in light yellow, and the major groove side is shown in orange. There's a greater distance between the phosphate backbone in the major groove than there is in the minor groove, and that's the biggest difference. Now, this has significance because DNA binding proteins will recognize the major groove and the minor groove and bind in a specific manner based on that conformation. Here we see a table that shows the organization of DNA in prokaryotes and in eukaryotes. Prokaryotes have a circular genome that's housed in the nucleoid, which is also called the nucleoid body. They also have circular extrachromosomal DNA called plasmids. Eukaryotes have a double membrane-bound organelle called the nucleus, where the genomic DNA is stored. This genomic DNA is comprised of linear chromosomes. Eukaryotes also have extrachromosomal DNA that's circular. This is found in the mitochondria, in animals and plants, and in chloroplasts in plants. The thing is, there's a lot of DNA that must be packed into a cell, Bacteria have on average 4 million base pairs of DNA per cell. This is approximately 1 millimeter long, but the bacterium is 1 micrometer long. That is a big difference. In mammals, it's even worse. We have over 2 billion base pairs of DNA per cell. And our DNA is 1 meter long, but the average nucleus is less than 10 micrometers in diameter. That's a lot of information to store in such a small space. So we package the DNA in several steps to condense it into this small space. First of all, we condense it through forming a helix, and then this helix is wrapped around a protein core to form a nucleosome. This nucleosome and its DNA form the 10 nanometer fiber, which is also called beads on a string, because that's what it looks like in micrographs. The 10 nanometer fiber can be compacted by winding it around to form a 30 nanometer fiber. This is a thicker fiber that contains more DNA in a smaller space. This DNA can be looped and joined to scaffold proteins in eukaryotic chromosomes. And this looped DNA can be wound even tighter to form highly condensed metaphased chromosomes. This is an electron micrograph of eukaryotic nuclear chromatin that shows the 10 nanometer fiber beads on a string. So let's talk about this beads on a string. It's formed of nucleosomes and intervening linker DNA. The DNA that's wrapped around the nucleosome consists of about 147 base pairs that wraps about 1.65 times around each histone core. The histone core itself consists of an octamer of 4 histone proteins, and each histone has a globular domain and an N-terminal tail. The N-terminal tail is actually a really important feature of the histone. Histones bind to one another to form a core particle that consists of 8 proteins. This is called a histone protein octamer. It contains 2 H2A, 2 H2B, 2 H3, and 2 H4. Now you can see that this octamer is found in the center and the DNA is wrapping around it. The amino terminal tails of the histones actually make contact with one another and contact with the DNA as it wraps around. This shows a nucleosomal core particle and the DNA wrapped around it. Histone tails are important because they contain lysine residues that bind to the phosphate backbone to anchor the DNA in place. The positively charged amino acids in the histone tails make salt bridges with a negatively charged DNA phosphate backbone, and this serves to anchor the DNA in place. Now the nucleosomes themselves become organized into a higher order structure called the 30 nanometer fiber. We see the 10 nanometer fiber on the left and the 30 nanometer fiber on the right. This 30 nanometer fiber is also called the solenoid. In the solenoid, the chromatin is highly condensed, but this means that genetic information is not readily accessible, not to replicational or transcriptional machinery, and this is fine if you just want to store the DNA, but if you actually want to use this DNA, the solenoid has to be unwound so that the enzymes and other proteins that function during replication or transcription can actually get access to the gene itself. And so, in many ways, the solenoid works like the rolling bookshelves that they have in some libraries. They come together and close down in order to store a lot of information in a small space. But when you want to access the books on these shelves, they open up so that you can have access. And a similar thing happens when the solenoid decondenses to allow access to the DNA that's wrapped around the nucleosomal core. Now the thing is, these solenoids, the 30 nanometer fibers, adopt this conformation because of interactions among the histones and between the histones and the DNA to form this higher order packing. Now, in order to get access to the DNA, these electrostatic interactions must be broken, and to do this, histone transacetylases add an acetyl group to the lysines in the histone tails to get rid of the positive charge on those lysines. The loss of this positive charge allows the solenoid to unwind. The electrostatic interactions between the histone core and the DNA that's wrapped around the core are actually reduced also through the action of histone transacetylase. And this again is because the positive charge on these lysines is no longer there, because there's now an acetyl group that's been added to that lysine. And in this way, histone transacetylases, abbreviated HATs, allow the DNA to unwind from the 30 nanometer fiber. This is sort of like the library bookcases opening up to allow access to the DNA for transcription. Histone transacetylases increase transcription because the transcriptional machinery can get access to the DNA, whereas histone deacetylases reduce transcriptional activity because the machinery can't get to the DNA. Histone deacetylases remove the acetyl group from the lysines and thereby restore the positive charge on the lysine to promote electrostatic interactions. And in this way, the 10 nanometer fiber becomes associated more strongly with the histones, and the 10 nanometer fiber condenses to form the 30 nanometer fiber again, to adopt a shut conformation, thereby reducing access to the DNA. In this way, histone deacetylases decrease transcription. And so this marks the end of Lecture 34. Have a great day.